Now today we will be discussing about bacterial structure in continuation with our previous video in which we have covered cell wall. In this video we will be discussing about cell membrane. So it is part 2. In part 1 we have already covered the cell wall part. Cytoplasmic membrane. In this video we will be discussing about the structure of cytoplasmic membrane, then function of cytoplasmic membrane, followed by a few MCQs for a quick revision. So here it is a morphology of bacteria. We have already discussed it. So we will just quickly go through it once again. In the morphology of bacteria, you can see there is an outer layer which is loosely bound. This is called as the capsule. And followed this is a cell wall. Cell wall is a thick outer covering, after which there is a periplasmic space. About cell wall and periplasmic space, we have already discussed in detail in part 1. Now today we will be discussing about this thin layer inside the periplasmic space. This is called as plasma membrane or cell membrane. This is invaginated at a place to form mesosome, and inside this plasma membrane is cytoplasm. Now within the cytoplasm you can see various structures like ribosomes. Ribosome is the machinery for protein synthesis. Then there is circular nucleoid, which is the genetic material of bacteria, and extra chromosomal genetic material which we call as plasmid. There are few cytoplasmic inclusions and few appendages. These appendages are called flagella or pili. Now coming to cytoplasmic membrane. Cytoplasmic membrane is also known as cell membrane or plasma membrane. These all words can be used interchangeably. So this cytoplasmic membrane structure was given by two scientists, that is as Jonathan Singer and Garth L. Nicholson. Hence it is also known as Singer-Nicholson fluid mosaic membrane model. This term fluid mosaic membrane model is very important because it signifies the structure of the cell membrane. They devised this, they proposed this cytoplasmic membrane structure in the year 1972. So what this cytoplasmic membrane consists of? Its constituents include lipids, proteins, and carbohydrates. These are the broad classifications. We'll go into the detail of each of these later on. And the thickness of cytoplasmic membrane is about 5 to 10 nanometer or 75 angstrom in thickness. Now different types of molecules in this plasma membrane, they are put together. Hence it is called as a mosaic. Mosaic means different crystals or glasses or pieces are put together to give a specific pattern. That pattern is called as a mosaic pattern. So similarly in this membrane, different molecules, that is the lipids and proteins, they are alternating and hence making a mosaic pattern. That's why its name is fluid mosaic model. Now why fluid? Because these molecules are constantly moving in two directions. The molecules, that is the proteins and lipid. These lipid and proteins, they make up the cell membrane, but they are not stiff or stagnant at a place. They keep on moving in two directions. They keep moving like a fluid. Hence it is called as the fluid mosaic model. Fluid because it is liquid nature, it moves, and mosaic because there are multiple type of proteins and lipids that are intercalating each other. Now we have already seen that the cell membrane is fluid in nature. So there are few factors or things that will affect the fluidity of this membrane. First, an important factor is temperature. Whenever the temperature of the environment is increased, the fluidity of the cell membrane will increase, because phospholipids stay apart in this condition. You can remember it like whenever we heat any saturated fat like a butter or a ghee, it turns more into a liquid form, its fluidity increase. Same way, since there are phospholipids, there is a lipid component in the cell membrane. On increasing the temperature, on heating it, the fluidity will increase. Second thing is cholesterol. Cholesterol, it regulates the fluidity. Sometimes it increases and sometimes it reduces. The details we will discuss later on. Third important factor is the composition of saturated and unsaturated fatty acids. So whenever the composition of saturated fatty acid is more, since they have a straight tails, they pack more tightly and hence the fluidity will decrease. On the other hand, if you have a look at unsaturated fatty acids, their tails have this kink or bend. Because of this bending, they cannot be tightly packed in the cell membrane and hence the fluidity will increase. So with saturated fatty acids, fluidity decreases because there is more tight packing of the membrane. In case of increase in unsaturated fatty acids, fluidity increases because there will be less tight packing of the lipid component of the cell membrane. Now coming to the detailed structure. It is also known as unit membrane and it is made up of phospholipid bilayer and proteins. Why this unit membrane? Because it is similar in all the organelles and any membrane of the cell. Not only plasma membrane, but if there is any membrane within the cell, like in organelles, they have a similar structure. It is called as a unit membrane. So this unit membrane mainly composed of two items, that is the phospholipids and proteins. Proteins make the bulk, that is 50 to 70% of the membrane, and there are about more than 200 types of proteins. So this is the major chunk of plasma membrane, that is proteins. The second biggest component is phospholipid, which constitutes about 30 to 60% of the composition. In case of prokaryotes, sterols is absent, but again there are few exceptions, like we have studied earlier mycoplasma. Mycoplasmas and ureaplasmas, when they grow in sterol rich medium, they can incorporate sterol, but otherwise by default there is no sterol present in the cell membrane of these prokaryotic bacteria. Okay. Now coming to the lipid. Lipid consists of three parts, three types, that is phospholipids, cholesterol or glycolipid. Here in this picture you can see this red colored lipid bilayer. This is phospholipid, which makes the major chunk or major bulk of the plasma membrane. Then this yellow colored structure which is embedded inside the phospholipid bilayer is cholesterol. The third one is glycolipid. Glycolipid means lipid is bound to carbohydrate. So this green colored structure which is present on the outer side of the membrane is glycolipid. So lipid is present in plasma membrane in three forms. One is phospholipid, which makes a bilayer, cholesterol embedded within the phospholipid bilayer, and a glycolipid where lipid is bound to. This is a lipid bilayer structure. In the cell membrane, there is lipid bilayer and intercalating proteins. So this lipid bilayer is also called as the phospholipid bilayer. It has two parts. When we have a look at the bigger picture, it has a head and a tail portion. Now in this head, there is a phosphate group and a glycerol group. These three carbon atoms, you can see this is the glycerol group, and at C3 there is attachment of phosphate group. Now this, because of the presence of this phosphate, it is also known as the phosphate head. Below this there are tails which could be saturated or unsaturated fatty acids. We have seen that saturated fatty acids are straight, unsaturated fatty acids have a bit of kink. Now this tail structure, this is hydrophobic in nature, means they are water heating. They stay away from the water, whereas this head portion is hydrophilic, that is they are water loving. They can stay close to the water. Since it is ionic and polar, it stays near the water, whereas this hydrophobic portion, it will stay inside the membrane. So here you can assume this is the exterior of the bacteria and this is the cytoplasm of the bacteria. But inside it, all the tails will align themselves to make a hydrophobic portion. Hence these molecules are called as amphipathic. Amphipathic means that have a hydrophilic and a hydrophobic part. Amphi means both. They like both water loving and water hating. Now coming to the membrane proteins. These membrane proteins are of three types, that is integral proteins, peripheral proteins and membrane associated proteins. What are integral protein? Integral protein are also known as intrinsic membrane proteins. These are amphipathic in nature, just like phospholipid. They are also amphipathic. That means they will have both kind of ends, a hydrophobic and a hydrophilic. So in the picture you can see this is integral protein. This is embedded in the bilayer matrix. So in this amphipathic protein, the hydrophobic domain is embedded in the hydrophobic part of the lipid bilayer. This is lipid bilayer and the hydrophobic part is embedded within this. Whereas it also has a few 1 to 2 hydrophilic domains. So these hydrophilic domains, they are protruding from the outside from the hydrophobic part of the lipid bilayer into the surrounding aqueous environment. So these hydrophilic ends are into the aqueous environment, whereas hydrophobic part is embedded within the lipid bilayer. These globular molecules are particularly embedded in the matrix of phospholipid, and the bulk of phospholipid is organized as a discontinuous fluid bilayer. This is discontinuous fluid bilayer. So these intrinsic proteins, they are bound to membrane by strong hydrophobic forces. Since these are very strong forces, they will need a drastic treatment with strong agents like detergents, bile acids or organic solvents to dissociate. So they will be dissociated, but they will interrupt or disrupt the membrane structure. So these are intercalated in the lipid bilayer, so they cannot be easily removed from the bilayer and need treatment with strong agents. Example of intrinsic proteins are integrins. So what is the function of these integral proteins? They are involved in structural integrity. Also, they act as channels, transporters, receptors and enzymes, and thus help in the transport of molecules across the membrane. Also, they are involved in cell signaling and communication between the two cells. That means the signal transportation and the communication between two cells, they help in that function. Moreover, few hydrophilic ion channels are formed by the assemblage of these transmembrane proteins. So these ion channels will be formed. Also, the cellular ionic balance and electrical properties of the membrane. These two things are also maintained by these integral proteins. So important functions are structural integrity. They act as channels, transporters, receptors. They provide ion channels. They maintain cellular ionic balance and also electrical properties of the membrane. So the second type of proteins are peripheral membrane proteins. What are these peripheral membrane proteins? You can see here these membrane proteins, they are attached by weak non-covalent or electrostatic forces. So since they are peripheral proteins, they are not strongly bound, they can be easily removed as opposed to the integral proteins. These proteins were very difficult to remove, but the peripheral proteins are easy to remove and they will be removed without destroying this membrane structure. So what is the function of these peripheral proteins? They provide membrane stability and curvature. They are also attachment points for various enzymes and signaling complexes. They are also involved in cell adhesion, maintenance of cell shape and recognition of self and non-self cells. So they have a role in immunity as well. The third type was membrane-associated proteins. These are basically transient interactions with membranes instead of intercalating into the lipid bilayer. So you can see these are glycoproteins. These are attached transiently to the cell membrane and they are not incorporated within the inside of cell membrane. They provide connections with other cellular components like enzymes, proteins or cytoskeleton elements and also help in structural integrity of the cell membrane. Now coming to the third portion, that is cholesterol. Cholesterol inserts into the lipid bilayer with its polar hydroxyl group close to the phospholipid head group. So this cholesterol also has a polar hydroxyl group. This hydroxyl group will be attached near the polar head of the lipid and the rest of the cholesterol will be embedded inside the lipid bilayer. So what is the function of this cholesterol? As we have already seen, we will be discussing it in detail. So here we discuss it. That is, it regulates fluidity. Now how does it regulate fluidity? When the temperature is high, what happens that cholesterol interferes with the movement of phospholipid fatty acid chains and making the outer part of the membrane less fluid. So whenever temperature rises, it does not allow this lipid layer to move a lot and hence it makes it less fluid, thus reducing its permeability to small molecules. So this helps in the selective permeability of the membrane also. So whenever the temperature is high, it will not allow the lipid molecules to move apart and hence reduce the fluidity. Whereas at low temperatures, cholesterol has opposite effect. What it will do? It interferes with the interactions between fatty acid chains and hence prevents the membrane from freezing and maintains membrane fluidity. So whenever there is low temperature, there are chances that the membrane may freeze. So what they will do? They will not let the lipid molecules interact with each other too much because they will come in between them and that's why the freezing will be inhibited. They are not present in bacteria, but they are a component of animal cell plasma membranes. Now coming to the last portion, that is carbohydrate. This carbohydrate in cell membrane is present either in the form of glycoprotein or glycolipid. Glyco means carbohydrate, protein is protein. So carbohydrate, when it is attached to protein, it is called as glycoprotein. When carbohydrate is attached to lipids, it is glycolipids. Here we have already seen this is glycoprotein present on the membrane surface. Another is glycolipid. This green portion which is present on the membrane is glycolipid. So they are present on outer side of the membrane as short branching chains attached to either protein or lipids. These are the sites on cell surface that allow cell to recognize each other. So they have an important role in. So carbohydrate coat covering the surface of the cell is known as glycocalyx. This glycocalyx is formed by oligosaccharides of glycolipids and transmembrane proteins. So glycolipids plus transmembrane glycoproteins, they collectively form a layer. This layer is known as glycocalyx. Now what is the difference between the cell membrane of archaea and bacteria? Cell membrane of some archaea contain unique lipid. This lipid is known as isoprenoid. Instead of the fatty acids attached to glycerol head, in the bacteria we have seen there was a phosphate head to which fatty acid chains were attached. But in case of archaea, there is a unique lipid which is called as isoprenoid instead of those fatty acids. And some of the lipids have no phosphate group, so they are not phospholipids. Another difference is that due to this unusual lipid content, they survive high salt, low pH and very high temperature conditions. So this difference makes them to survive in these harsh conditions also. So let's discuss the summary of the structure we have studied so far. So we have seen that cell membrane is made up of components like lipid, protein and carbohydrates. Lipid include phospholipid, which is the major chunk of the membrane. It is a bilayer in nature and amphipathic, means hydrophobic as well as hydrophilic, both parts are there. Another lipid is in the form of cholesterol, which is present between phospholipid bilayer and it regulates the fluidity of the membrane. Then thirdly, it is also available in the form of glycolipids, which is attached to carbohydrate moiety. Another component is protein. Proteins can be peripheral proteins, integral proteins or membrane associated proteins. Then there was a carbohydrate component. This carbohydrate component can be bound to protein to form glycoproteins or it is bound to lipid proteins. Now let's see the functions of semi-permeable membrane. This membrane regulates the permeability and transport of solutes across the membrane. This membrane is known as semi-permeable or selectively permeable because it allows the entry and exports molecules outside the membrane selectively. Few molecules are allowed while the other are not. Another function is electron transport and oxidative phosphorylation. Also, it is involved in excretion of hydrolytic exoenzymes and pathogenicity proteins. Moreover, it is involved in biosynthesis of various structures we will be discussing in detail, and it has some chemotactic function. So the first function is permeability in transport. This is the most important function of cell membrane, because of which it is also called as semi-permeable or selectively permeable membrane. Why selectively permeable? Because it allows entry and export of selected molecules only. It will not allow all the molecules to pass through it easily. So it allows selective permeability and transport of solutes. It makes a hydrophobic barrier around the cell. Because of the lipid bilayer, there is a hydrophobic barrier. So it needs a various transport processes to transport the solute. What are these transport processes? They could be passive transport, active transport, group translocation or spatial transport process. Let's see what all these transport systems mean. Passive transport is diffusion. Diffusion means there is no energy is needed and the solute at higher concentration will move from outside to inside at a lower concentration. So since this is simple diffusion, there is no energy involved, so there will be no speed and no selectivity. Example is oxygen, carbon dioxide and water. Here you can see the solutes are in higher concentration, so they will move from higher to lower concentration by simple diffusion, and this process just takes place because of the difference in the concentration on the two sides of the membrane, and no energy is required for this process. This is a passive process. This is not active process. In active processes we need energy. So in passive process, there is no energy, hence solutes will move from higher concentration to lower concentration. Now there is another method of passive transport, that is facilitated diffusion. Again, in this kind of diffusion, there is no energy is required, but it is selective because it is occurs with the help of few channel proteins like glycerol. So there will be few proteins like transmembrane proteins that will allow the movement of solute from outside to inside or inside to outside. So this is not energy dependent, but it is selective. This process is quite uncommon in prokaryotes. Now coming to active transport. Active transport, as we have already discussed, it takes the use of energy. So it can move on the opposite basis, means nutrients can be concentrated manifold. Even if the concentration is low outside, still the nutrient can move from low concentration to high concentration. Why? Because we are providing energy for the transport. So on the basis from where this energy is derived from, it can be ion coupled transport or ATP binding cassette transport, also known as ABC. So let's see what are these two. Now what happens in ion coupled transport? The transport happens because there is some ion gradient across the cell membrane. So whenever there is an ion gradient across the cell membrane, that gradient will be utilized to create some energy, and that energy is used to transport the molecules even if it is at lower concentration outside. This type of transport is common in aerobic organisms, and the force that is required is known as proton motive or sodium motive force. It can be of three types. It can be uniport, symport and antiport. So we will have a look at what thing. Now coming to uniport. Uniport catalyzes transport of a single species independent of any other species. Suppose this is outside of the cell, this is inside of the cell. So there is only single kind of molecule which is being transported in a single direction from out to in. There is no other molecule involved and no other direction involved. This kind of transport we call uniport. Single species independent of any other species. In case of symport, there is co-transport, means there is transport of two kinds of dissimilar species but in the same direction. So these two dissimilar species are being transported in the same direction, but they are two dissimilar molecules. There is no single kind of molecule. This type of transport we call as symport. Lastly, we have antiport. In antiport, there is exchange transport of two similar solutes in opposite direction. These are not same, but they are similar in nature, and they are getting transported in opposite direction. Like the dark blue circle, they are transported from outside to inside, and the light blue one, this is being transported from inside to outside. And this type of transport is seen in approximately equally in 40% of the times. Another method was ABC, that is ATP binding cassette transport. This transport system uses ATP to transport solutes. So this is facilitated by binding proteins. There are few proteins in the plasma membrane which are involved in this kind of transport, and it occurs at the expense of ATP. So whenever the protein transfer the substrate, they transfer it to the membrane bound protein, and there is hydrolysis of ATP. When ATP is hydrolyzed, we know that energy is generated, and that energy is utilized in opening the pore inside the membrane. So when the pore is open, the substrate can go inside. So this is unidirectional kind of flow. Now group translocation. What happens in group translocation? It is also called as vectorial metabolism. So this transport is basically coupled with some kind of metabolism, like it happens in case of glucose. What happens when the glucose is transferred? It will be metabolized. So it picks up the glucose, but then it phosphorylates it during the transport, and sugar phosphate is released into the cytoplasm. So some kind of metabolic changes are occurring in the molecule, and instead of the same molecule, a modified product will be delivered. Then there are special transport processes where the internal compartment of the animals we have seen, they have no free iron. It is mostly bound to proteins like lactoferrin and transferrin. Hence some bacteria secrete siderophores. Siderophores are the items that will chelate iron, and they help in transport of the iron as a soluble complex using membrane proteins. Second function was electron transport and oxidative phosphorylation. Now what happens here? There are various cytochromes and other enzymes like dehydrogenases of the respiratory chain that are present in this membrane. So it is kind of a functional analog of mitochondrial membrane. Because of these cytochromes and various enzymes, electron transport and oxidative phosphorylation, respiratory chain will happen on the cell membrane. Excretion of hydrolytic exoenzymes and pathogenicity proteins. So organisms that are dependent on polymers like proteins, polysaccharides or lipids for their nutrients. What they do, they release some hydrolytic exoenzymes. So they will release some enzymes that will break these polymers, and these subunits will transfer penetrate the cell membrane. So whenever the organism they are dependent on polymers like proteins, polysaccharides or lipids, they release few enzymes through the membrane, and then these will break these polymers into smaller molecules that will be transferred. Bacteria secrete them into the external medium either directly or through periplasm. Biosynthesis. Now the cell membrane has carrier lipids on which cell wall subunits assemble and cell wall enzymes are synthesized. Phospholipid synthesis enzymes are also located on the cell membrane. So this kind of synthesis and synthesis assembly takes place on the cell membrane. Chemotactic function. Attractants and repellents bind to the cell membrane. They are called as chemoreceptors. Now, what are mesozymes? We have seen in the structure earlier that at some point the cell membrane is invaginated into the cytoplasm to increase the surface area available for various cellular enzymes. Also, it helps in DNA replication. Though some studies suggest that they are just artifact, but most of the investigations show that they are just there to increase the surface area for the cellular enzymes. Now coming to the MCQs. First question is, which of these is not a constituent of cell membrane? A. Phospholipid, B. Glycolipid, C. Glycoprotein, D. Lipoteichoic acid. A. Phospholipid, B. Glycolipid, C. Glycoprotein and D. Lipoteichoic acid. So the correct answer is D. Lipoteichoic acid. We have studied in cell wall. So this is not a component of cell membrane. Now coming to the second question. The structure of plasma membrane includes a lipid layer between protein bilayer. So they are saying that there is a single lipid layer between protein bilayer, or a lipid bilayer with transmembrane and peripheral proteins. So they are saying that there is a bilayer of lipid along with transmembrane and peripheral proteins. C option is a lipid bilayer surrounding a protein bilayer. So there are 4 layers here. Then a D option is a lipid bilayer with lipopolysaccharide on the outer surface. So what is the answer? Answer is B. That is, there is a lipid bilayer with transmembrane and peripheral proteins. We have seen that in the center there is a lipid bilayer and transmembrane and peripheral proteins are embedded in that lipid layer. Next question. Lipid molecule in cell membrane are arranged in. What is the pattern in which lipid molecules are arranged? Is it scattered, B. head parallel, C. alternate, or D. series? The answer is head parallel. So heads are aligned and the tails are aligned and they form the bi. Next question. Ion coupled transport is a feature of. We have seen there was ion coupled transport. It was seen in cell wall, B. capsule, C. cell membrane, or D. all of the above. Answer is C. cell membrane. Question 5. Which of these is not a function of plasma membrane? A. Transportation of solutes, B. Provide rigidity to cell, C. Oxidative phosphorylation, and D. Excretion of exoenzymes. This is a bit tricky. Answer is B. Rigidity to cell. So we have seen that providing rigidity to cell is mainly a function of cell wall, whereas the other three options are more apt to cell membrane. This is the main place where transportation of solutes take, oxidative phosphorylation and excretion of exoenzymes takes place. Thank you. If you like the video, please share it and subscribe to the channel.